Hello and welcome to BMI 4302 Virology. This course is brought to you by the Department of Microbiology, Faculty of Biotechnology and Biomolecular Sciences, University Putra Malaysia. I'm Dr. Eddie here. Today we would like to talk about the enveloped positive-stranded RNA virus. In previous lectures we talked about the naked positive-stranded RNA virus, and today we are going to look at the enveloped one. Right, and that will be the first part, and after that we will talk about the life cycle of a selective virus. In this case we have chosen dengue virus, where we look at the detailed life cycle of the virus. Of course, the 6 steps: the attachment, penetrations, translations of viral proteins, genome replication, assembly, and egress or release. Without further ado, let's start with the first part, the introductions of enveloped positive-stranded RNA virus. So we talked about this in the previous lectures, and where we chosen the Picona viridae as a selective family of virus to look into detail, specifically on the poliovirus, where we study in detail how the poliovirus attach and penetrate, as well as the life cycles. Alright, and these 3 family of the viruses are mainly naked viruses that cause human diseases, and we also going to look at we also talk about like 3 other families which are enveloped viruses that cause human diseases. So the one that we are going to focus on is the Flaviviridae, a family that composed of viruses that cause like dengue, yellow fever, JE, hepatitis C, and so on. Alright, I'm sure everyone will be like looking at, wow, why don't we talk about coronavirus? Well, dengue virus is one of the diseases. Dengue virus is one of the viruses that cause dengue around the world, and in fact, it is a very, very important disease, especially on a tropical country. So in 2005, we only have 500,000 of cases, and now in 2019, as of last year, the cases has increased up to 4.7 million cases. So even though the death cases is not as much like coronavirus, but I'm sure once you understand the life cycle, once you learn how the virus infect, replicate, and so on, you can replicate the understanding that you learn from Flaviviridae into Coronaviridae. Alright, so let's have a look at family of Flaviviridae. So it has 4 genera. It used to have only 3, which is Flavivirus, Pestivirus, and Hep C virus. Recently, Virus has been reclassified as a new genera. So now, Flaviviridae have 4 genera. The Virus used to be under Hep C virus. The genome of these 4 genera are similar in size and organizations. Let's look at this is the genomic organizations. So what we can see here is the structural protein, which is located at the 5' end, and non-structural protein is located on the 3' end, because this is a positive-stranded RNA. So you have the structural on the 5' end, non-structural on the 3' end, and you can see it's quite standard. You have the capsid on the far left, then with the enveloped protein next to it, and then followed by all the non-structural protein, where they are protein that is important for the virus replications. So the virus genome is more linear, single-stranded, and positive strand or positive sense means that the virus RNA is positive strand and single-stranded, and more importantly, they are monopartite. What it means is like the RNA is not segmented into a few segments. It's just one long linear RNA. Of course, it's a single ORF, means that you only have one start and one stop codon, whereby the start codon start from here all the way and here. So it's a single ORF, just one reading frame all the way to the end. So what it means also is that the protein will be the viral protein will be translated from beginning until the end as a long polypeptide and then clipped into individual protein for their functions, similar to the one in poliovirus. Okay, so I'll let you to draw your attentions to these particular structures here. This is for flavivirus genome, and flavivirus genome is the only general in Flaviviridae that has cap, and whereas Hep C, Pesti, and Pegivirus, all three of these genera have IRES, and they are 5' end. All right. So the flavivirus are composed of viruses that cause dengue fever, yellow fever, JE, Japanese encephalitis, and you can see they are quite serious disease that caused by this particular family, encephalitis. Yeah. So pestivirus, on the other hand, are causing diseases in swine and cattle, and Hep C is the one that causes hepatitis C in humans, and Pegivirus is the one that used to be called hepatitis G or GB virus that infect the human and chimps. All right, and what we can see from here, transmitted method by these viruses for flavivirus are mostly mosquito-borne or insect-borne or tick-borne, whereas the Pesti on the contact, and hepatitis C and Pegi on the parenteral and transfusions. What it means by parenteral is it means that you have to break through the skin and allow the virus to transmit into your body, parenteral. Okay, so this, don't confuse this with mother and fetus transmissions. Parenteral means that you break the skin for the transmissions. Alright, what about the disease that apart from the flavivirus that I mentioned earlier on? So hepatitis C causes liver cancers, but for Pegivirus, it has been all along quite persistently infecting human, but yet there is no known disease that caused by this particular group of virus yet. Maybe one day in the future we'll know. Alright, so when we try to analyze the NS3, the non-structural protein of the whole group of viruses, what we can see is that we can build a phylogenetic tree, and what we can see here is all of these are flavivirus. Then we separate them into Pegivirus and Hep C virus as well as Pestivirus. And you can see from earlier on, this tree has been branched out from the flavivirus. So it's quite obvious that they are not within that same clade. So over here, what we can see also is when you build the phylogenetic tree, you can see the group that are closely related are on the vector base. For example, this group of viruses, they are tick-borne flavivirus. This group of viruses are mosquito-borne viruses, and this group of viruses are insect-borne. It occurred to us that the virus also evolved according to the vector, where the way they transmit also they also affect how the phylogenetic tree is being built. Alright, so the one that we are interested in to look into detail for this flavivirus will be dengue fever or dengue virus. So let's have a discussion on what's dengue. I'm sure everyone knows about what dengue is. So the dengue is caused by dengue virus. In short, we put DENV, alright, DNV, which stands for dengue virus. There are 4 serotypes, serotype 1, 2, 3, and 4. Now the differences between these 4 serotypes is about they have similarity of 65 to 70% similarity. Alright, so the this is the virus that causes dengue fever, dengue hemorrhagic fever, and dengue shock syndrome. Alright, so if you are bitten by a mosquito, Right, say for example, this is the first time you got bitten by this mosquito, and not all the mosquito carry dengue virus anyway. But let's say you're so unlucky that you got bitten by this mosquito that somehow carry the virus. So this virus go into your body, and what happened next is like, because your body has not exposed to the virus before, so your body started to react, and your immune system start to fight off the virus, and basically you'll be recovered from that. So for primary infections, people who are just first time exposed to this dengue virus, basically they are asymptomatic, meaning that they will not develop dengue fever or whatsoever. However, once the person has been infected with, let's say we use dengue virus serotype 1 as an example. Let's say the person has been infected with dengue virus serotype 1. What happened in their body is that they started to develop immune system against dengue virus serotype 1. Okay, so what happened is that next time when he got infected, got bitten by another mosquito again, which carry dengue virus serotype 1, so he's basically, or she's basically safe from the virus. However, the dangerous part of this is that the dengue virus serotype 1, because they are quite similar to serotype 2, 3, and 4, up to 65 to 70% similarity. So what happened next is the antibody that being produced that's supposed to neutralize dengue virus serotype 1, it somehow partially neutralized serotype 2, 3, and 4. And what dangerous is, how dangerous it is when a person got exposed to dengue virus serotype 1, and when he got exposed to dengue virus serotype 2 or 3 or 4. Okay, let's use 2 as an example. So when this dengue virus serotype 2 being introduced or being transmitted into the patient or the person, what happened is this antibody will partially neutralize the virus. And we know that when your immune system is working, it will bind to the virus and neutralize it and prevent it to cause any infections. However, because it's just partially neutralized, means that the virus has not been fully neutralized, they are not fully inactivated yet. What happened is they can easily escape and cause infections. So what happened now is during the secondary infections, so this red color is supposed to be antibody that neutralize your serotype 1, dengue virus serotype 1. So now they partially neutralize serotype 2. And what happened next is when this antigen is bound by antibody, and it will bind to the Fc receptors on the APC, antigen presenting cell, or monocyte, macrophage, dendritic cells, and so on. So these are the cells that will attack the antigens and antibody bound complex. So what happened here is the Fc of the antibody will now bind to the Fc receptors on the APC, which will then bring the whole virus or complex into the cell. So this process we call antigen dependent enhancement. So it enhances the binding or the entry of the virus into the APC cell. And what happened here, because it's partially neutralized, virus can escape, and now what happened? The virus started to replicate inside the macrophage. Alright, so now, what happened now? So previously, if you have a primary infections, the virus takes time to find their whole cell that is suitable for them to attach to and go into the cell and so on and so forth. But now you have this antibody dependence enhancement, enhance the virus to go into the host cell, and this resulting in the virus to replicate even more inside the host or inside the person. So now what happened next? So this is just a hypothetical diagrams. When we say someone who have dengue, I'm sure everyone knows that check whether he has rashes over his body, if he has dengue fever, and so on and so forth. So what happened here? Now we explain that later. So what happened here is when the virus go into your body, this is a secondary infections, right? So what happened is your immune system will react, and then they will release some cytokines to alert your body that now we are under attack, so immune system has to work. So in the meantime, because this virus has been exposed, this person has been exposed to the virus before, but on a different serotypes. And what happened is your immune system, your B cell, will started to produce antibody against the dengue virus, even though it's not the the correct or the accurate one. So this antibody that being produced, it will be dengue virus antibody, like antibody against dengue virus. Alright, so now back to the statements that when you have dengue fever, you might have rashes all over the body. So what happened is that there is a cross-reactivity of this antibody that being produced, and this antibody started to attack the epithelial cell. So we know that our circulator, our blood vessel is layered by a single layer of vascular cell, vascular endothelial cell. This is one layer of cell. Alright, this is your blood vessel. Okay. So what happened here is when the antibody started to attack your endothelial cell, this made the endothelial, the vascular become leaking, and which allowed the blood to leak out from your blood vessel. Okay, so blood vessel supposed to be layered by endothelial cell, which is quite impermeable to blood. All right. However, because the attack of the antibody making this endothelial cell or your blood vessel become leaky, so now the blood can leak out, and that's the reason why you can see spot of red all over the body, because the blood has escaped from the blood vessel and go under your skin, and then you can see leaky spot all over the body. Alright, so that's one phenomenon. What about the second phenomenon that we say, oh, the platelet has reduced, drop, it's quite dangerous. What happened here is the antibody also cross-react with the platelet. And we know platelet is very important in our body. Platelet is the one that once it's activated, it will cause will clot the blood or whatever and so on. So however, the antibody that being produced now attack the platelet. Okay, so this is sort of like autoimmune symptoms where your own antibody attack your own cell. So what happened here is that your platelet is being attacked and being killed. That is the reason why we have thrombocytopenia, meaning that you have a reduced number of platelet. So this is one of the reasons why when we talk about dengue virus, we say the platelet has been reduced, and it's getting reduced and reduced. It's very dangerous and so on and so forth. Of course, we also cannot deny that there might be some complement cascade that play around with that play a role on the decay of the blood vessel. Alright, so now we have explained dengue hemorrhagic fever. So what about dengue shock syndrome? Like I mentioned earlier, the leaky blood vessel, if this continue, then you have more blood and leak out from the circulatory system. We know that our blood circulatory system is a closed system where the heart pump and blood go through, artery, go to your whole body, capillary, then back to vein, and so on and so forth. So it's a closed system. So your heart pump, and then it has certain pressure. That's why you check your blood pressure. It has like 120 to 70. So what does a shock mean? A shock in your body is not electrical shock, but a shock of your body is when your blood pressure has fall below the normal. It becomes so low that your body do not have enough of blood that circulate in your body, and your body enter a shock. So how does this eventually become a dengue shock syndrome? It's because the blood vessel becomes so leaky, and the heart try to pump harder so that the blood can go through whole body and come back to the heart. But because it become leaky, so imagine you have a balloon. When you press, all the air go out, and so eventually you do not have enough pressure to push it forward. So that is making your body become a shock. So dengue shock syndrome is quite dangerous, which might lead to death in the patients. Right, knowing that the dengue virus is quite dangerous and quite quite common in tropical country, and now we have 4.7 million cases as of last year. So scientists are trying to develop, a lot of pharmaceutical company also trying to develop vaccines. And what we can see here, we have several types of vaccine that are present here. We have live attenuated vaccine. Well, first of all, we have Denvax, which and TB003 and TB005, which are now in clinical trial phase 3. So remember we mentioned that if you want to create a vaccine for dengue, you must have the 4 serotypes. You can't just use one serotype as vaccine. That is the challenge when you want to develop vaccine for dengue virus. So in this case, what does a live attenuated vaccine means is the virus are still active, but they have been weakened, meaning that the virus, even though they can go into your body, they can still replicate, but they has been weakened in such a way that they are no longer causing any disease in your body. Alright, so this is what you mean by live attenuated vaccine. So you can see that replacing the coding sequence of DAN2 vaccine with DAN134. So what it means is that you have all 4 dengue virus serotypes in this vaccine. So the second type of vaccine is inactivated vaccine. Alright, so these vaccines basically is the virus, but they use chemical to inactivate it. For example, in this case, they use formalin to inactivate the virus. And then this vaccine will contain 4 serotypes of the virus, and then inject into the patients and let them expose to it and develop immune system against the virus. Then we also have subunit vaccine. Now what the subunit vaccine means that you don't need the whole virus to become a vaccine. You just need portion of the virus that you know exactly where the portion is important that induce immune response. So you can just use that part of the virus and become a subunit vaccine. So it's actually recombinant truncated protein contained than ATE. So it's just a portion of the virus. Then we also have a DNA vaccine, means that you just put DNA that contain the virus sequence. So then your body will produce the protein, and this automatically protein that being produced will recognize by your body as foreign antigens. So your body will react and produce immune system against it. And last, we have heterologous prime and boost. Basically, this is just a method of creating the illicit immune response in your body. So it started with TLAV. What it means is that you have all 4 serotype that is live attenuated virus. So they use live attenuated virus as the first injection to your body. Then they boost it with PIV, purified formalin inactivated virus. So basically, it's just a combination of this and this to create a vaccine in the patients. So all these are in clinical trials. We are quite hopeful for the phase 3 vaccine because we need to overcome this problem, especially for us in the tropical country. Every year we can hear people suffering from dengue. Right, let's look at the virus that causes dengue, the dengue virus. Right, so dengue virus is a positive sense single-stranded RNA virus that has a genome of 10.6 or 10,600 bases. Right, 1 open reading frame, the one that we mentioned earlier. Alright, so it has 3 structural protein, where the structural protein is the capsid that's surrounding the RNA. You have a precursor M or pre-M and envelope, which is on the surface of the virus. Then we also have 7 non-structural protein, which is NS1, 2A, 2B, 3, 4A, 4B, and 5. So just if you want to remember, the even number have A, B, the odd number doesn't have A, B. So this is how the structures of the virus look like. So on the surface, the yellow color that you can see here is the envelope protein, and the orange is the pre-M or M protein. And because it's an envelope virus, so definitely it has a lipid bilayer. The blue color here is the lipid bilayer that derived from the host. So the host membrane has the envelope and M protein on the surface. Going in, we can see the icosahedral structures, which is the capsid protein. But unlike poliovirus, you have VP1, 2, 3, 4. This is only just one type of protein, one type of capsid protein that form the icosahedral structures here. So going inwards into the core of the virus, what we can see here is the genomic RNA of the virus. Alright, so the structure is relatively simple. So I'd like to draw your attention here is when we look at the structure of the virus, so the diameter of a matured infectious virus is about 50 nanometer. Okay, it's about 50 nanometer from tips to tips. However, when the virus is inactive, all right, or immature, it's normally about 60 nanometers. And what you can see here, the difference between this one and this one is quite smooth and this one is spiky. So later, when we look at the life cycle of dengue virus, we'll understand why this becomes spiky and so on and so forth. Alright, so that's it for our first part, right? For the introductions of envelope positive strain virus. Hope you enjoy it. Thank you.